So thank you very much for introduction, and thank you for this opportunity to present you our work comparing brain metabolism and edema in chronic hepatic encephalopathy in developing an adult rat brain in Vivo and Longitudinal study. I have no conflicts of interest, so I would start with short background. We know that in acute form of hepatic encephalopathy in adults, there is brain edema caused by high ammonium delivery to the brain and subsequent glutamine accumulation with no time for secondary osmotic regulations. We also with antioxidant problems and with systemic and neuroinflammation with neurotransmission impairments and astrocyte injury. In children, symptoms are very similar to adult one with also brain edema and astrocyte injury, but ammonia induces irreversible changes with inhibition axonal growth and subsequent cell death. And this damage depends on maturation stage of the brain and how long and strong ammonia exposure is. And how I said, unfortunately, these changes are irreversible in comparison with adults where a lot of neurological symptoms seem to be reversible when ammonia levels return to normal values. In chronic liver disease in adults, there is a spectrum of cognitive impairment, starting with minimal hepatic encephalopathy, and molecular basis is up to now unclear, but it's well accepted that ammonia is the main toxin and astrocytes are the main target with Alzheimer's type 2 astrocytosis, and also with the evidence of low-grade brain edema at the end, and with possible contribution of cholestasis. In children, chronic liver disease has a lot of questions up now, but it's already proven that neurocognitive impairment, which occurs before liver transplantation, persists also after. And even patients with non-cirrhotic portal hypertension have impaired cognition. So based on this already known difference in adult and developing brain in acute form of liver disease, our hypothesis is suggesting different susceptibilities to chronic cholestatic liver disease according to age. And our aim, using great advantage of high-field magnetic resonance spectroscopy and diffusion, so it's to elucidate metabolic changes of the developing rat brain in chronic liver disease and how these mechanisms differ from those in adult rats. Our methods. To study chronic liver disease, we use bile-aggregated rats. We are studying evolution of illness of the same animal during 8 weeks, scanning them every 2 weeks. Pups have 21 days on the day of bile-aggregation, and we chose this age because development stage of rat brain of 21 to 30 days corresponds to human brain in childhood. We are using 9.4 Tesla variant scanner for MR spectroscopy and diffusion tensor imaging. Also plasma measurements for each scan for measure ammonia, glucose, bilirubin and other metabolites. Open field behavioral tests for motor activity. At the end, histological measurements of the brain for GFAP and water channel aquaporin 4. And also absolute water content with a wet and dry method. So with high-field MR spectroscopy, we can see more than 20 metabolites in the brain, and these are markers of myelination and cell proliferation, energy metabolism, osmoregulation, neurotransmitters, and antioxidants, and all this in vivo and non-invasively. Our results, so the localized brain spectroscopy were acquired from hippocampus, and here you can see 3 different spectra. So this one is normal portion spectrum from healthy sham-operated rat, This one is from adult BDL rat 8 weeks after BDL surgery, and the same from developing brain. And already visually it's possible to see increase of glutamine and diminution of myoinositol in BDLs. And if we have a look in more details, we can also visually see difference between developing brain and adult brain. So now more precisely, if we have a look on the evolution of glutamine during 8 weeks, so this is concentration of glutamine measured by MR spectroscopy, we can see progressive increase reaching threefold physiological value in PAPs next to only twofold increase in adults. And this increase in glutamine is causing secondary osmotic imbalance, and myoinositol is the small light showing the most prominent decrease, and it's already shown on the previous slide, reaching 30% fall in adults and 50% fall in developing brain. And well, but this bigger drop is expected as a compensation of higher concentration of glutamine in developing brain. Also, all other metabolites are showing most prominent changes in PAPs, showing that PAPs are more sensitive to chronic liver disease. So there is decrease in neurotransmitters, glutamate and aspartate, 20 and 40% in PAPs, next to 10 and 30% in adults. Also in decreased antioxidant capacity of brain cells, represented by glutathione. So 30% significant decrease in developing brain, next to only 9% non-significant decrease in adults, and also 40% increase in lactate in PAPs, next to just 10% non-significant decrease in adults. For brain edema, we can see increase of apparent diffusion coefficients in PAPs, 30%, and only 10% in adults, and together with increase of absolute water content, there we can suggest there is a brain edema. And PAPs are reaching also higher values of ammonia and bilirubin in plasma at the end of the illness in comparison with adults. From immunofluorescence, so here we have images from PAPs. These are from adults. On the top row there is GFAP, and here is aquaporin 4, and we can see astrocyte swelling. From GFAP we can see astrocyte swelling in both PAPs and adults, and from aquaporin 4 we can see that there is higher increase in aquaporin 4 expression in PAPs in comparison to adults BDL. And at the end, the behavioral results. So these ones are for adults. So this is distance moved and time spent in movement. So we can see a significant decrease of motor activity already 6 weeks after BDL. In adults, and the preliminary results in PAPs are showing the same tendency. And to conclude, so we are for the first time showing that PAPs with BDL show greater osmotic and metabolic changes in the brain than adult rats. And so PAPs show significantly stronger osmotic changes, decrease in neurotransmitters and antioxidant capacity, increase in lactate, for edema, increased apparent diffusion coefficient, aquaporin 4 expression, and astrocyte swelling. And for behavioral results, similar tendency in decrease of locomotor activity as adult rat. And how this contributes to neurocognitive changes remains to be determined. I would like to thank my supervisor, Kristina Kodelbu, and all other colleagues contributing to this project, and thank you for your attention.